Hello, welcome back to a new recording of an SDP project. So today I am exploring two different targets. My first target is how to make and defend a claim based on evidence that inheritable genetic variation may result from genetic combinations through meiosis. So my explanation is that genetic variation happens through meiosis because of how the egg and sperm cells divide and share DNA from each partner. This results in every offspring to be a slightly different version of both parents. Although if closely related offspring breed through multiple generations, they might develop genetic diseases that are less common in offspring that breed with organisms containing vastly different traits from them. According to a website called Science Learning Hub, during fertilization, one gamete from each parent combines to form a zygote. Because of the recombination and independent assortment in meiosis, each gamete contains a different set of DNA. This produces a unique combination of genes in the resulting zygote. Recombination, or crossing over, occurs during prophase 1. Homologous chromosomes that are inherited from each parent pair along their lengths, gene by gene. Breaks occur along the chromosomes as they are joined, trading some of their genes. The chromosomes now have genes in a unique combination. My second target is to create and defend a claim based on evidence that inheritable genetic variation may result from errors during replication. Inheritable genetic variation may result from errors during replication because of the many types of gene mutations that can occur during meiosis and mitosis. There are a multitude of different mutations that can take place during cell replication, especially in the DNA replicating process. According to the well-established article paper by the U.S. National Library of Medicine, there are 6 main types of genetic mutations during the replication of the cell. The first is the missense mutation. This type of mutation is a change in one DNA base pair that results in the substitution of one amino acids for another. The second is the nonsense mutation. A nonsense mutation is also a change in one DNA base pair. Instead of substituting one amino acid for another, however, the altered DNA sequence prematurely signals that the cell to stop building a protein. The third is insertion mutation. Insertion changes the number of DNA bases in a gene by adding a piece of DNA. As a result, the protein made by the gene may not proper functionally or at all. Deletion. Deletion changes the number of DNA bases by removing a piece of the DNA. Small deletions may remove one or a few base pairs within a gene, while larger deletions can remove an entire gene or several neighboring genes entirely. The deleted DNA may alter the function of the resulting proteins. Duplication. Duplication results and consists in a piece of DNA that is abnormally copied one or more times. This type of mutation may alter the function of the resulting protein. And the last type of DNA mutation is called frameshift mutation. This type of mutation occurs when the addition or loss of DNA bases changes a gene's reading frame. A reading frame consists of 3 groups of bases that each code for one amino acid. A frameshift mutation shifts the group of these bases and changes the code for amino acids. The resulting protein is usually non-functional. Insertions, deletions, and duplications can be all classified as frameshift mutations.